We begin with a controversial contagion study during the Spanish flu, which was considered highly infectious and from which more people are said to have died than in the entire previous First World War. In this study, attempts were made to infect healthy people in various ways by sick people, but this simply did not work. In other words, a real contagion experiment on humans under extreme conditions, which has never been duplicated and published in a comparable way even to this day, more than a century later. We then explore in detail why more and more people are certain that the SARS-CoV-2 virus has never been isolated, or for that matter, that it even exists. Statements that even most unvaccinated people and critics of restriction measures consider absolutely preposterous. Nevertheless, we will try to understand how this view is reached. To do this, we look at what virology has been publishing since 1952 as so-called virus proofs and how computers are used to calculate the genetic information of the supposed viruses. We will also explore how crucial control experiments for the detection and disease-causing properties of the particles known as viruses were virtually never conducted until 2021, with a few overlooked exceptions in the 1950s. The control experiments mentioned in passing at the time and in detail since 2021 now shatter the entire narrative because they completely refute the crucial underlying assumption of proof. Let's start with an American study from the year 1918, when during the so-called Spanish flu, epidemic contagion experiments were carried out on 62 volunteer healthy naval sailors. Among others, Johns Hopkins University was already involved in these extensive experiments at that time. Various contagion attempts were made to spread the pathology of the Spanish flu influenza, which was regarded as highly contagious from sick to healthy people. To that end, various swabs and secretions were taken from the diseased individuals, such as mouth, nose, throat rinses, coughed up mucus, and blood draws. From this supposedly highly infected material, secretions were then administered to the volunteers in a variety of ways. Dripped into the nose, sprayed into the throat and eyes, or injected under the skin. Was infection possible in this way? No. To the great surprise and puzzlement of the researchers, the whole thing had no significant effect at all. After all these attempts to force infection on the 62 volunteers failed and no influenza symptoms could be induced, experiment number 7 used a considerably more harmless approach, one in which infection is assumed and claimed to occur to this day. For this purpose, 10 volunteers sat down opposite 10 patients in succession, talked briefly, and then allowed themselves to be coughed on directly several times. Each of the volunteers were thus directly coughed on by 10 different sick people. Again, none of the test subjects experienced any symptoms of infection as a result. In 1918, it was still technically impossible to see particles the size of so-called viruses, since the electron microscope was not invented until the 1930s. The idea that previously invisible particles could cause disease was thus a pure hypothesis ever since Louis Pasteur in the 19th century, a claim which could only have been technically verified or falsified much later. So for decades, it was actually impossible to detect particles the size of so-called viruses in a direct way. But nevertheless, the infection theory advanced in those days from a theory to a generally accepted truth, without any proof. Today, direct virus detection would be possible, and almost all people assume that this has been done thousands of times, only this is not the case. Therefore, we now briefly look at how a disease-causing virus could actually be detected relatively easily and without any doubt. We then compare this with modern virology and its criteria for an assumed detection. Using the example of SARS-CoV-2, anyone can, with reasonable effort, analyze the few available studies in which the virus was supposedly isolated at the onset of the pandemic. In fact, such isolation has never occurred, nor has it occurred with any other supposed virus to date. The problem is the use of the word isolation. These dozens or more studies talk about isolation, but contrary to the meaning of the word and what people commonly believe, it is not isolation and never has been. Because according to the Duden Dictionary, isolate means something that usually occurs only in conjunction with something else, separate from the other for a specific purpose and presented in a pure form. What should it look like? Let's start with what a real isolation should look like with logical common sense. If you suspect a new virus and want to detect it, you should take samples from as many people with the same specific symptoms, in addition to further samples from a healthy control group. According to virus theory, the assumed viruses multiply inside the host cells and then leave these cells, so they must also be amply present outside these cells and therefore be contained in the samples. No other substances that might contain their own genetic code should be added to these samples, otherwise it would not be possible to distinguish later whether the observed particles or genetic components came from the additives or from the original sample. These samples are then split, crushed, and filtered as precisely as possible so that only the very smallest components are present in the desired dimensions. Since the suspected viruses should also be present outside the cells, in principle only the filtering step should be sufficient, without prior lysis, that is, without dissolving cell membranes. The filtrate would now be ultracentrifuged, that is, rotated extremely fast so that the components are arranged according to their size and mass. This is a common technique to then observe and analyze thousands of particles of the same size and shape under an electron microscope. In this spectrum of different particles, a subset would now be assumed to be the viruses responsible for the disease. These suspicious particles should not be present at all in the samples of the healthy control group, or if so, then in much smaller quantities. The purity of these particles would be tested and ensured so that they could subsequently be further characterized. For example, the genetic material could be extracted directly from the purified particles and published as the real genome of the suspected virus. By definition, viruses are exogenous, that is, they come from outside the body and are supposed to cause disease. They are not endogenous, part of the body, where they serve some other, as yet unexplained, meaningful purpose. To prove that these particles are really so-called viruses, then, they would have to be transferred to healthy subjects where they would presumably multiply and subsequently lead to the same symptoms or disease. And finally, they would also have to be isolated from samples from the newly sick test subjects in the same way. This is precisely what is described in Koch's famous postulates and is also communicated accordingly as the only sensible course of action. Of course, here again, a control group is necessary in which a placebo is transferred and from which no symptoms should occur. Nowhere near any of this has been done for SARS-CoV-2, let alone with any other so-called virus claimed to cause disease. However, with other particles of the same size, such as exosomes or so-called bacteriophages, exactly this kind of approach to isolation has been done for many years to characterize them. So it is a standard procedure that works. In virology, on the other hand, this real directed detection has never been done. Instead, they proceed in a completely different way. To understand this procedure, let us look at the two common variants. 1. How the very first so-called isolation and discovery of the viral genetic material occurs. And 2. How all subsequent studies and scientists isolate the virus and orient themselves to this protogenetic material, also what they accept as proof of the supposed disease-causing properties. We will look at the first case using the example of SARS-CoV-2, namely the very first study from China in which the supposed genome of the virus was decoded. According to the current scientific consensus, a genome is the totality of the genetic information of a living being or supposed virus. From a single person with lung problems, a sample is taken from the fluid collected from a pulmonary irrigation, in English abbreviated as BALF. This sample is now broken down into tiny components. The huge amount of gene segments found there are extracted, prepared using a wide variety of potentially error-prone steps, and then subjected to genetic analysis. What does this mean? RNA and DNA as carriers of genetic information consist of so-called nucleotides as building blocks, which in turn each contain, among other things, one of 4 specific bases, shown here in red, green, orange, and yellow. A DNA or RNA strand is characterized by the sequence of its different base pairs. Genetic analysis of all gene segments found includes determining the sequence of bases, which is called DNA sequencing, with the aim of characterizing the complete genome of the virus. From this large set of now sequenced gene segments, called reads in the studies, the researchers exclude those which are found to be similar to known human gene sequences. All others are assumed to the presumptive virus, completely ignoring the possibility that the sample may contain a variety of other microbes of known and unknown species, whose genetic material is also included in the mixture. An attempt is now made to assemble a kind of puzzle out of all these tens of millions of gene segments by putting them together in an overlapping manner, where each piece would fit an enormous number of other pieces. There are now millions of calculated possible outcomes of the puzzle called contigs. And depending on the computer program and set parameters used for this step, completely different results can emerge. In this study, the programs Megahit and Trinity were used, with the latter giving more than 3 times as many results. This alone shows how arbitrary the findings are when completely different results are produced with different software. So which result is real for the virologists and could present the genome they're looking for? The result of the sought-after puzzle, that is, the complete genome or the complete genetic material of the supposed virus, is at this point unknown. However, one now simply suspects a similarity to another virus previously discovered in the same manner, that is, to another genome previously generated on the computer, and therefore one determines the similarities to all other genomes present in the database. In this case, the greatest similarity was found to an assumed harmless bat coronavirus, and this was then simply used as a template for the puzzle. The principle of the procedure perhaps becomes more clear if one imagines the patient's sample like a gigantic puzzle collection of tens of thousands of puzzle boxes, each puzzle representing real genetic material. One suspects an unknown puzzle in this collection, which, based on the scientific consensus to date, is estimated to be responsible for the disease which afflicts the sample's owner. An obvious procedure would be to sort the puzzles according to suitable criteria and to take all puzzles eligible by size and determine by further experiments whether one of them is indeed the cause of the puzzle owner's illness. These would be the steps of a real isolation, as well as Koch's postulates, but they are never performed. Instead, all the boxes are opened and all the puzzle pieces are dumped onto a gigantic pile. For all these tens of millions of pieces, images are now created on the computer. Then you remove all pieces that have a similarity with pieces of harmless puzzles from known manufacturers, which are already stored in your database. In doing so, all manner of mistakes occur. If, for example, in a known puzzle, a single colored piece of a certain shape is stored, all pieces of this color with a similar shape are removed, even if there are corresponding colored pieces in almost all known puzzles. So after this step, you probably won't be able to completely solve any of the original puzzles. But there are still millions of pieces left. From these remaining pieces, you now try to solve the unknown puzzle, even though there are still pieces from thousands of puzzles here. Since you don't know the puzzle and its meaning, you more or less ignore the picture on the pieces and only try to find shapes that can be joined together, as if you were to flip all the puzzle pieces upside down and see which ones can be somewhat joined. Now there are hundreds of thousands of ways to put puzzles of different sizes together, and depending on the settings and the type of computer program used for this step, you will get wildly differing results. This means that the results are in fact a collection of purely fictitious puzzles which have never been seen before and do not exist. Still, the question is raised, which of them is the one we're looking for? For this, one compares all these fictional results with the database of all other puzzles stored as disease-causing, which were also all previously similarly calculated. This comparison yields the most similar pair, which is now designated as the solution. This puzzle is a pure computer simulation and did not previously exist in the prior puzzle collection. It is a purely random composition of pieces that have nothing to do with a real puzzle. This puzzling is called sequence alignment, in which different DNA regions are compared to identify similarities between the identified gene segments and the template. All open gaps or missing puzzle pieces are then simply filled in by a computer algorithm. Consequently, millions of puzzles or genomes could then be assembled from the extracted material in the same way. Simply by finding the best match to a template, a new genome can then be determined by computer and auto-completed. This more or less random calculation has now been added to the international database as the genome of SARS-CoV-2. This genome does not exist in reality. A virus with this genome does not exist in reality. At no time is an actual virus isolated in the sense of the word and confirmed by electron microscopy. At no time is a genome determined from a real, actually existing particle. At no time is a real existing particle known to be causative of disease. In fact, this first study does not even claim that the alleged virus, which is supposed to be part of the computer-generated genome, is really the cause of the disease, but only that appeared in the context of the disease. Nevertheless, Christian Drosten published his PCR test based on this genome in the database within days after it became known. This was before the previously discussed study with the genome construction and without proof of a causal relationship was even published. The PCR test is able to determine in what quantity certain gene segments of this non-real calculated total genome are present in a dissolved patient sample. In our puzzle comparison, this means that the PCR test checks whether 3 specified pieces of the puzzle are contained in a gigantic pile of puzzle pieces. If so, this is taken as evidence that this exact puzzle was present in the puzzle collection. That these matching puzzle pieces can be found in small quantities in every pile of millions of puzzle pieces is not mentioned. Only when a certain arbitrarily defined quantity is reached is it claimed to be proof that the puzzle you are looking for is indeed there, making the test positive. This critical amount above which the PCR test is considered positive is regulated by the CT values. There are no uniform regulations for the parameters used in this test, and only very rarely is public information available. Above a certain value, virtually every test is positive. So if a person tests positive with symptoms, he is considered to be infected. If a person tests positive without symptoms, he is also considered infected. And all because fragments of nucleic acids were found there, to which a corresponding dangerous meaning was attributed on the basis of a computer generation of a fictitious genome without any scientific basis. That is, without any proof that they really occur in a so-called virus, or that this unproven virus is actually responsible for the disease. This type of PCR test is now used in all further studies claiming the isolation of SARS-CoV-2. From the beginning, it was the only argument for the apparent presence of a pandemic, because in 2020, the number of people who fell ill or died was not higher than usual. It is only because of the PCR test that huge numbers of symptomless people were counted as infected. This is the reason why many people who have studied the test and its development in detail have always spoken of a test pandemic. So far, then, we have a fictitious virus that has never been isolated and for which no causal link to the disease has been established. In addition, we have a computer model of a genome, which also does not exist in real life, and a PCR test based on this model. All other existing studies, which now detect the same virus, now proceed more or less in the following like manner. First, we look at the principal steps in the diagram, then on the basis of one of the many existing studies. Samples are taken from a few people with or without symptoms, but with a positive PCR test. A tissue culture, for example from monkey kidney cells, also called vero cells, is starved in order to add the allegedly infected material. This presumably allows the supposed virus to multiply there. Antibiotics are added to this culture to exclude effects caused by bacteria. The sample is now added to this tissue culture and then further starved, disintegrating into all sorts of particles and dying cellular components. Contained viruses are simply assumed to be the cause of the die-off, the so-called cytopathic effect, and are apparently verified accordingly with the PCR test. This is seen as proof of the virus, although cell death can, of course, also have completely different reasons, as we will see in a moment. It continues to be referred to as virus isolation, although this has absolutely nothing to do with isolation in the sense of that word. On the contrary, this soup now contains all kinds of genetic material from various sources, from the human, from the air previously breathed in by the human, from the monkey cells, possibly the added substances, etc. It is now again subjected to a genetic analysis, as already described earlier, from which a more or less similar genome is calculated. The difference to the genetic analysis of the very first study is that they now already know what they're looking for. The fictitious genome of SARS-CoV-2 is now used directly as a template for the sequence alignment. Of course, it is possible to generate a puzzle from the millions of puzzle pieces, which almost completely match the fictitious puzzle that we're looking for. If individual puzzle pieces should actually be missing in the gigantic pile or are not considered similar enough, they are simply filled in artificially. If the differences in the final puzzle are too great, the genome is interpreted as an alleged mutation or variant. So what we see here is classic circular reasoning. For a hypothetical virus, which is assumed to be the causative agent of a disease, a hypothetical genome is generated on the computer based on the template of another hypothetical calculated genome, which is then taken as evidence that the virus exists and is responsible for the symptoms in humans. A PCR test is created that can detect minimal real-world parts of this fictitious whole genome in samples. In samples from humans where the test was positive, an infection with the hypothetical virus is now supposedly proven by attributing the cell death in the test tube to it because the PCR test apparently confirms its presence. A genome is then again calculated from the extracted material, this time using the SARS-CoV-2 template. Depending on how similar the new calculation is, it is either a known variant or another hypothetical mutation of the hypothetical virus each time the differences in the calculation exceed a threshold. So we see that this approach never actually isolates a virus and certainly never shows that there is a causal relationship with any disease as a result in the sense of Koch's postulates. In some of these studies, photographs of the hypothetical virus are shown. In any case, it can be seen that these labeled particles are never present in isolation, and thus something is simply labeled which is thought to be the virus. However, this could just as well be exosomes or any other extracellular particles in the same order of magnitude. Or even artifacts under the electron microscope, which, according to Dr. Harold Hillman and Dr. Stefan Lanka, are the result of massive chemical and mechanical manipulation of the material. In order to know that a particle is a so-called virus, it must first be isolated, transferred to another human or animal, and cause the same disease there as described in Koch's postulates. Otherwise, it is just something not specified. As in any other scientific discipline, it should nevertheless be self-evident that for all these steps, at least control experiments should be made. That is, that the same experiment be done with samples from healthy people and no cytopathic effect with death of cells occurs, since this is considered evidence of the virus. Or also the same experiment without adding a sample. Or in the case of SARS-CoV-2 with a sample of another familiar lung disease. Or also whether various other virus genomes, for example, of HIV or measles, can also be constructed on the computer from the soup by simply using their templates when puzzling. All these mandatory control experiments were never done, or at least never published. Even without adding samples from diseased persons, the alleged gene sequence of SARS-CoV-2 can be constructed from the gene fragments contained in the soup, just like the gene sequences of HIV, measles viruses, and others. This means that the gene segments found are not components of virus material foreign to the body, but rather completely normal components of the body that are ejected from the cells in the course of the experiments and were merely misinterpreted as viruses on the basis of decades of incorrect theory. With the refutation of virology, the entire vaccination dogma would, of course, also be refuted. Because where no disease-causing virus exists and where a genome was only conceptually generated on the computer, all PCR and rapid test procedures based on it, as well as vaccinations, are, of course, simply consequential errors without any sense or benefit. Of course, all this sounds completely unbelievable. However, a first check is fortunately very easy. Therefore, let's now take a look at one of these studies from the beginning of 2020 from Germany as an example, which claims the isolation of SARS-CoV-2 and implements exactly the previously described procedure. In this case, material was taken from a single test person who had no symptoms, but only a positive PCR test. After all, the term asymptomatically ill has only been used since the PCR test was given a corresponding meaning. The sample was added to monkey kidney cells, so-called viro cells, mixed with antibiotics, as well as depriving the cells of much of their nutrition. After 4 days, the cytopathic effect began, that is, cells died. Done. So where is the real isolation of a virus, as suggested in the title, from which one could determine an actual genome? Where is the filtering, the purification, the centrifugation, etc., described here? Where is the control experiment to prove that the cytopathic effect would not have occurred without adding the sample? Subsequently, exactly the same procedure for calculating the fictitious genome is once again described. To the knowledge of the critics, a real infection in humans has not succeeded in scientific experiments, neither at the time of the Spanish flu nor to this day in other experiments. The control experiments, which could disprove the misinterpretation that assumed viruses in the sample were responsible for the death of the cells, were never carried out except for brief mentions in the 1950s. It was not until 2021 that Dr. Stefan Lanka had them performed and showed that it was not supposed viruses that were responsible for cell death, but the experimental setup itself. No virus has ever been correctly isolated and proven by means of Koch's postulates or adapted Rivers criteria to have pathogenic properties. Instead, the word isolation has been redefined in virology, and indirect effects have been equated with isolation. In the case of SARS-CoV-2, as with all other viruses, the genome was computer-generated using so-called sequence alignment, and according to the control experiments of Dr. Stefan Lanka, could be generated in the same way from completely healthy endogenous material. In the same way, the genomes of hundreds or thousands of other believed variants and mutations were generated on the computer. It will probably not be long before someone, somewhere, again interprets the harmless endogenous particle as a dangerous virus. A new genome will be generated on the computer, new PCR tests will be developed for it, and another test pandemic of who knows what size will occur.